you very much, Mr. Chair. First of all, I'd like to thank the organizers for inviting me to give a presentation, and I think this is a wonderful forum where One Health meets. Having said about One Health, I think many speakers since yesterday have enlightened the importance of One Health. After two elegant speakers, the Director of Research of Karnataka Veterinary Animal and Fishery Sciences University and my predecessor here who spoke before me, Dr. Sri Krishna Islur, it is only in the fitness of things that we now move on from an environmental hazard to an animal hazard in the context of One Health, and now on antimicrobial resistance in One Health, but with a focus on aquaculture, which indicates the role of antimicrobial resistance with specific, I should say, importance to the aquatic environment, aquaculture. We all believe that water is the elixir of life, and any organism or plant grown in the water naturally is most important, therefore, but very often water becomes a victim of all of the pollution that is taking place, including antimicrobial usage in every sector, finding its way as residues or as antibiotic resistant determinants to the aquatic environment. And therefore, while it tends to be used in aquaculture also for the treatment of diseases, it also means that we need to be very aware of the difficulties and of the problems associated. And we need to take care because a One Health approach has to be looked at all angles. Earlier it was a tripartite way of looking at it, the human, the animal, and of course the plants. But now, apart from WHO, FAO, and the World Organization of Animal Health, you have the United Nations Environmental Program being signatory to looking at addressing this important aspect of One Health and thereby contributing to the Sustainable Development Goal. Climate change, urbanization leading to more people moving in, a lot of travel with people moving across globe, and of course companion animals have all made the problem even more important than ever before. We know that many contributions to global threats are there. It could be diseases, non-communicable diseases like cancers and diabetes. You could also have a lot of communicable diseases, the diarrheal illnesses, cholera, and you could have diseases, others like road traffic accidents. But if you see, the most important prediction made is about antimicrobial resistance in 2050, which would be probably a pandemic. That's what it would be called, where over 10 million deaths costing about 66 trillion pounds is the estimate. So the problem is more serious than what is imagined, and the rise and fall of antibiotics, very explicitly explained in this book, makes very interesting reading. We know from the history, from the time the antibiotics were discovered, we find that there has been a lack of development of antibiotics, probably because we have run out of all of them in our ability to isolate, and secondly, I think the pharmaceutical concerns have lost their interest in developing new antibiotics because the microorganisms are outplaying them each time, with resistance developing at a very rapid rate when you look at the deployment and the resistance, which is why one is looking also at alternatives, which are also something that I will be speaking about. When penicillin was first discovered, there was a euphoria because we had a broad spectrum compound that could help. And I'm sure Alexander Fleming, sir, had been very excited about the discovery. But that was not very long-lived, because he also made this statement, quote, "The thoughtless person playing with penicillin treatment is morally responsible for the death of the man who succumbs to infection with the penicillin-resistant organism. I hope this evil can be averted." So it's kind of prediction even by the person who first discovered antibiotic. And if we look at the 10 years challenge that is shown, look at the way the sensitivity to organisms was observed, even in a conventional method such as the Kirby-Boardis method, and then we see in 10 years time resistance, and those showing even small zones are in fact resistant, because we know that just because there's a zone seen, we do not say it is sensitive, because the blood level concentrations attained by a particular antibiotic. Many of the presentation figures and all would be from my own articles in, of course, journals where the impact factor has been very high. What I want to say is it's not just about a high impact factor, but about the impact that such articles should have and the way forward by researchers to look at the various aspects. And water, as I said, receives effluents from hospitals, pharmaceutical concerns, all anthropogenic waste, yes, then from the poultry, the piggery, the livestock farms, and then you have the human gut serving as a pot boiler where the resistance gets transmitted to other organisms that are present there. And then, of course, many mechanisms by which the transfers happen. So there is a reservoir of antibiotics, and what is happening is an enrichment that is taking place even in the aquatic environment, in the wastewater treatment plants, and therefore the resistance is transmitted in several ways. And World Health Organization has, of course, identified this as one of the important global threats, and the United Nations General Assembly in 2011 adopted this political declaration on antimicrobial resistance. World over, this has become a hot topic to speak about. This is just to show how in different sectors the names of the antibiotics used for the different bacterial pathogens. As we know, antibiotics are mainly targeted to bacterial pathogens, but very often empirical treatment due to clinical presentations of different kinds of biotic agents causing the problem leads to, I should say, misuse of the antibiotic. It happens in human health, it happens also in animal health, in aquaculture, which is no exception. Now, if you look at the names of the antibiotics, they are very familiar to every clinician, every laboratory worker present in this hall, and we will see the same is seen in the aquaculture sector, whether it is for shrimp or for fish. The bacteria may be varying, but they may be either gram-positive or gram-negative, and if you look at the antibiotics that are used, they are the same. What is used also in poultry for the pathogens encountered there. Then the question arises, why should we speak about resistance? It's just as important as, say, some cardiac problem. We train people to resist chocolates, fat substances to eat and all. Same way here, the problem that is going to be associated with the use of antibiotics needs to be minimized. We don't say it should not be used, because it needs to be used whenever there is a bacterial problem, but how judiciously we use it. And in sectors such as aquaculture, it is very often an extrapolation of what has happened, what happens in human health or what happens in animal health. But in aquaculture, there is a difference. We realize that the transmission routes have to be understood very well. A lot of the antibiotics have to be added either to the water or to the feed. It can't be directly finding its way into the animal through an injectable unless it is a brooder, which is a very large-sized fish or a large-sized shrimp. But most often it is just added and finds its way to the waters where it acts on all the innocuous organisms taking part in the biogeochemical cycles. So this is just a presentation, but it's not so bad. Nobody need hesitate so much to eat. I'm a vegetarian, so I hesitate a little bit looking at this picture. But if we look at various sectors, we find aquaculture probably receives less than all the other sectors where it is shown. So if you look at the piggery, poultry or cattle sector, there's a lot more that gets to be used. And the mechanisms remaining the same, we will see that the antibiotics also therefore target the organisms that you suspect is the agent causing a particular problem. There are many resistance mechanisms, and therefore if you isolate an organism which is resistant to tetracycline from a human waste, or you isolate it from an aquaculture environment or from a poultry, to also see whether it is the same organism, because backtracking and trying to find out the source is as important when we talk about pollution that takes place. We find that the mechanisms can sometimes tell us whether it is the same organism that has actually been moving or not. Of course, NGS, and then looking at the resistomes and where it's located, studying all of the genomic methods of analysis of the microbiome to look at resistance factors is the way forward. But you can even look at mechanisms of resistance to, in a broad way, say whether it is the same or not. A caution about antimicrobial resistance. Now, there is intrinsic and acquired resistance. Why we talk about intrinsic is, we must remember not to use the antibiotics to which an organism is intrinsically resistant, because antibiotics were never made by the organism to save the humans. They were prepared for their own function, either structural or any other functional or metabolic function. We have used it for our benefit. Of course, acquired one is when through mutations or through horizontal transfer, such as transduction, transformation or conjugation, it spreads to others. Now, the intrinsic resistance to a particular antibiotic, a good example is Aeromonas, which is now recognized also as an important human health pathogen. You will see that this has intrinsic beta-lactamases which make them resistant to penicillin, and therefore the study of intrinsic resistomes and acquired resistance is very important. In the context of aquaculture, I take a few examples. Viable multi-drug resistant bacteria have been cultured from a cave in Mexico, which was obtained more than 4 million years ago, much before antibiotics were discovered. Marine bacteria found as far as 522 kilometers offshore at depths were resistant to antibiotics, and therefore it clearly shows that evolution of antibiotic resistance predates the evolution of actinomycetes. So antibiotic resistance genes have not evolved to protect against antibiotics. So these are some examples of resistance genes. About the beta-lactamase we spoke. Efflux mechanisms have basically been developed to push out all of the toxic substances such as pesticides, heavy metals, etc., which may be found in the aquatic environment, and this happens even when an organism acts the same way on an antibiotic that we try to use to cure a particular infection, but it may be just using the efflux mechanism to push the antibiotic out. So there are also metabolic functions, as I mentioned before. So there are many factors, and I made a brief mention about how they form part of the gene pool. Now, what will replace a failing antibiotic is very important to know. Vaccines have conventionally been used, but more recently we know about the antimicrobial peptides generated from stem cells, immunotherapeutics, then we have the CRISPR-Cas9. Many of these were spoken in the yesterday and this morning, and we have others such as probiotics, then bacteriophage therapy, the fecal microbial transplant, nanobiotics. Now, why are we looking for alternatives? Because in the aquaculture environment particularly, there is emergence of resistance, buildup of the pathogens, chemical residues in the shrimp meat or fish meat, and antibiotics used destroy the useful organisms in the environment. Their effectivity in seawater is questionable. Many antibiotics are actually destroyed by the cations that are present in the waters. Chemicals have been therefore overused to compensate the ineffectivity, which is very bad, and environmental deterioration happening due to the residues is a very important point. Now, probiotics is a term we are very familiar with. The prebiotics and probiotics together now are the symbiotics that are used, and of course our review papers in most of the journals give a lot of details, but I'm just covering very briefly for want of time, only just leaving, because there are a lot of young people who will take back message to work on certain areas. So prebiotics are the food for the probiotics, and then we have them together given usually when effectivity is to be best obtained. So you can have the inhibition of pathogens, which will result in a reduction in the use of antibiotics, and then a consequence would be the reduced antibiotic selection pressure, and therefore finally mitigation of resistance. So this happens through various mechanisms. It could be reducing the pH in the colon, production of antimicrobials, inhibition of colonization, and then of course the abundance of beneficial bacteria. This is very important because when antibiotics are used, they destroy not just the pathogen, but they also destroy a lot of the normal flora, which results in actually some of the survivors of the antibiotic treatment overgrow because the microbial balance is disturbed. Whether in the gut or in the water, the story is the same. And so one of the more recent ways in which we have been approaching is bacteriophage therapy, for which of course the government of India gave the Technology Development Award to us, and in the aquaculture environment it is used very much. And there are lytic and lysogenic, so we need to do whole genome sequencing to look at whether it's a lytic one, does it carry resistomes in it, because that would be paramount to deciding if a phage is going to be useful for sustained use in the aquaculture environment. And then there are many factors in an aquaculture environment. The human cells as well, though it's not yet approved in India. Those of us who have read the book, The Perfect Predator, would know how important this is a life-saving method. And in the case of aquaculture, we need to look at the salinity of the waters, depending on whether the fish is grown in fresh water or marine waters or estuarine waters, the temperatures at which they are grown, the cold waters or the warm waters, the pH of the waters, and of course total dissolved solids, in order to see how much of the phage gets actually disturbed by the presence and becomes ineffective. So there are of course vaccines for the 21st century, and we have been successful in having nano vaccine developed in our own laboratory, probiotics developed, apart from publication, technologies which have gone to the farm as well. And more recently, this has been a very important area for many people working on phytochemicals. Mr. Chair, can I take 1 minute? The last two slides. So we will see that the antibacterial activity of many phytochemicals is due to either the leaf extract or the stem extract, the essential oils in them, alkaloids, but most importantly, we find that the terpenes and the polyphenols, they are highly active against microorganisms. And where do they act? It's mostly on the bilayer, the cytoplasmic membrane, cell membrane, and the mechanism could be by their insertion in the bilayer or the potential alteration, their protein binding abilities, or increasing the permeability. Bacteriocin is another area, the antimicrobial peptides that we have developed both from the invertebrates as well as from other microorganisms. Niacin is something we're all very familiar with use in food safety, but we work on other bacteriocins as well, which have been developed from the probiotic organism, so that can we replace the organism by molecules that may not be amenable to resistance because they are very small antibiotics, small antimicrobial peptides. And this is another very potent area. And of course, last slide is about the integration of all of this, as we see all forms of life in the center, and we find that if we have to decrease antimicrobial usage, there are many aspects of research that have to contribute. Okay, it starts with breeding, whether it is in aquaculture, livestock, poultry. Humans, we don't call it breeding, of course, but in all other sectors, we have to have genomics coming in. Gene editing technologies are being used increasingly, microbiota modulation through targeted sequencing, and then we have the bacteriophage therapy about which I spoke. The antivirulence strategies, quorum quenching, happening usually through phytochemicals or certain other molecules, and then last and the most important preventive strategy, that is vaccination. Of course, CRISPR-Cas9 is being used to target the resistant organisms and to manage antimicrobial resistance, in which we also have a project which we work on. Okay, thank you very much for the opportunity.
Thank you, Doctor. Thank you very much for your